So I think we can all agree that we have one of the most beautiful coastlines in the world. And we can also be quite proud because we have one of the most well-protected coastlines in all of Africa. So many of you may know that we recently implemented 20 new marine protected areas, or MPAs. However, even with these new MPAs, we are only at 5% coverage of our EEZ. So we're only halfway to our 10% goal by 2020. So we got our work cut out for us. But I would argue that we have our work cut out for us now more than ever, because now within conservation planning, we have to account for this. So this figure is basically showing the annual global temperatures from 1850 on the left to 2017 on the right. And so I really wanted to include this figure because I think it really nicely portrays the climate crisis that we are currently in. So when we think about conservation planning for the future, we have to think about three main things with regards to climate change. Number one being climate velocity. So that is how much and how fast the climate is going to change. And then there's different ways that species can respond to climate velocity. So they can either shift their distributions or they can stay put and adapt. And one of the best ways that we can predict how species may change their distributions or adapt into the future is actually by looking into the past. So now I want to take us back roughly 21,000 years ago to the last glacial maximum. So things were a little bit different back then. The temperature was roughly 4.5 degrees cooler, and the sea level had dropped drastically, minus 120 meters. However, there were certain areas that remained relatively more stable over time since the last glacial maximum until now, and we term these areas climatic refugia. And there's been a few very nice studies that have looked at the relationship between sea level variability and climatic stability since the last glacial maximum and its relationship with genetic variation. And genetic variation is important for that feature that was species adapting, because if you have higher genetic variation, you should have higher resilience to future change. So this is just a nice example from Toms et al. 2014, where they identified two refugia. Let's see, is this going to... Okay. But two refugia in the green and the pink on the map at the last glacial maximum due to sea level change, and it nicely correlated with two genetic lineages of a rockfish. And then here's another example from Yannick et al. 2014, where they looked at climatic stability in the Arctic and compared it to genetic diversity of a caribou. So I just want to point your attention to the bottom right-hand graph. So we have climatic stability on the X with haplotype diversity on the Y, which is a genetic diversity metric. So we see there's a positive relationship. And this makes sense because a more stable area should harbor larger population sizes, which leads to higher genetic diversity. But as of yet, no one has actually looked at the relationship between both sea level change and climatic stability with genetic diversity of rocky shore species. So that was what I aimed to do within the first chapter of my PhD. So briefly, I wanted to identify both past and future climatic refugia for 3 rocky shore species, and then test the correlation between sea level variability and climatic stability with genetic diversity, and then to identify conservation areas based on these analyses. To do this, I had 3 study species, namely the granular limpet, the common shore crab, and the Cape urchin. And we chose these species because their distributions cover a range of environments, and also because they have different life history characteristics, and the genetic data was already available for all 3 species. Here is the current distributions of our study species with these are the presence data that I obtained, and then this was the genetic sampling sites from previous studies. For my methods, I first had to calculate climatic stability, and to do this, I ran species distribution models, or SDMs, and the predictor variables for these models were both air temperature and sea surface temperature, and this was available for 1,000-year snapshots back 21,000 years. So I first downscaled the environmental data with Craigie interpolation, and then I ran 21 models over the 21 time steps back in time. And each model outputs a habitat suitability map. And each cell in the map has a habitat suitability of 0 to 1,000. And then what I did was I just summed the habitat suitability per cell over the 21 models to get climatic stability for each species. Then I calculated sea level variability by looking at the Euclidean distance between the 0 and minus 120 bathymetry lines in QGIS. And then the genetic data was already available, from which I calculated nucleotide and haplotype diversity in DNA SP and Arlequin. Then I ran linear models to compare these three features. So looking at climatic stability since the last glacial maximum until now, we see here, so this is the suitability over the 21 models, with areas in red being highly stable over time. And then you can see that there are certain areas that were red for each three species. So then the bottom left-hand map, the areas that are in red there are the ones that had a habitat suitability of 800 or more at every time step for all three species. So these are the shared climatic refugia. Then when we look at the relationship between climatic stability and genetic diversity, what we're seeing here is on the x-axis, we are going from least stable to more stable over time. And then the top row is haplotype diversity, and the bottom row is nucleotide diversity. And basically, we see that there pretty much is not a significant correlation, except for what we see with nucleotide diversity with the crabs. And this is really interesting because you may remember in the graph with the caribou, we would expect, or what I would expect, is a positive relationship. But instead, we see a negative relationship. And it's basically the same story when we look at sea level variability. So just note here, our x-axis is going from more stable to least stable, so increasing in variability. So we would expect to see a negative relationship, but instead, we either see no significant relationship or a positive relationship for the crabs with nucleotide diversity again. So this leads me to ask the question, is sea level variability and climate stability even what's the main drivers of genetic diversity in rocky shore species? And I would argue that it probably is other forces that are the main drivers. So it could be the margin effect, which says that within the current distribution, the core should have higher genetic diversity than the range edges. It also could be due to contemporary environmental or biological factors, such as selection, or also due to admixture between historically separated populations. So say we had two lineages that broke off and then they got back together and interbred, and that area of interbreeding should have higher genetic diversity. So I'm currently exploring these in additional chapters, but now I want us to halt here and fast forward into the future. As you may remember, one of my aims was to identify future refugia. So now I ran species distribution models into the future with two time steps at 2050 and 2090, which was a combination of air temperature for 2070 and sea surface temperature for 2100. And here I'm just going to show the intermediate emission scenario, RCP60. And again, I ran SDMs with Biomod 2 and R. So when we look at 2050, we see that the amount of suitable habitat has decreased for all three species. And then the shared climatic refugia in the bottom left-hand corner, we only have a few cells on the south, southwest, and west coast. In 2090, things look even more bleak. So most of the eastern edge of the distribution is now uninhabitable. And our shared climatic refugia for the three species is only on the west coast and southwest coast. But what I think is really interesting is when we look at the shared climatic refugia throughout time. So these are areas where all three species were predicted to be highly suitable from the last glacial maximum predicted to 2090. And what's really interesting is two areas pop up as being climatically stable over time, mainly around False Bay and Cape St. Francis. And this is really interesting because the study by Toms et al. 2014 highlighted these same two areas as refugia. And then on top of that, the False Bay region has also been recently proposed as an EBSA area, meaning that it's ecologically important. And on top of that, when we look at the number of range-restricted endemic marine invertebrates, so this graph here is from Ewald et al. 2012. And on the x-axis, we have the distance around the coastline. And then on the y, we have the number of range-restricted endemics. And those two spikes correlate with False Bay and Cape St. Francis. But when we look at the current MPA network, these two areas remain unprotected. And so that's why I would argue as we increase our protection from 5 to 10% in the future that we should consider these two areas because they've been shown to be biologically, ecologically, and now evolutionarily important. And with that, I'd just like to say thank you, mainly to my supervisor, my funders, my lab, and if you want to hear more, you can follow me online here. Thank you.